Okay, man, finally I'm back, ready to continue, very eager to continue forward. I know I'm a little bit behind, but I'm going to try to catch up as best I can. So let's get into today's discussion. I am excited, actually. This is I think the most interesting, most biologically significant macromolecule. So I know it's been a little while, but if we can put ourselves back in the context of macromolecules, big molecules made by dehydration synthesis. We're taking small little units, monomers. We talked about the amino acids for proteins. So we take amino acids, connect them together to form the primary structure, secondary structure, tertiary structure of those proteins. We looked specifically at enzymes, which are a subcategory of proteins, little catalysts. We know that they have optimal temperatures, optimal pH. They can be inhibited. So that's information, that's knowledge that you should already have committed to, to long-term memory. So we're going to move onwards today to cover nucleic acids. So another big molecule, very significant, very important molecule in biology. So nucleic acids are specialized for the storage and transmission of genetic information. So this is what links basically the child up to their parents, the maternal lineage, the paternal lineage. This is what connects you to your offspring. If you have children or you're going to have children, this is what connects one branch of the family tree to the other. This is what we can analyze. These are the instructions that we inherit that make us look the way we look, that convey your, your skin color, your eye color, your height, whether you're going to be good at this or be good at that. All of these are coded for by the nucleic acids. Right? And there's two categories, two molecules that fall within this broader umbrella of nucleic acids. So we're going to look at DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So these are the two. There's a bunch of different types of proteins. There's only two types of nucleic acids. So a little bit structurally simpler, but much more critical. I mean, the proteins are going to help to construct us, build our ligaments, build our muscles. The DNA is what conveys the instructions on where to put that DNA or where to put those proteins, those muscles, those connective tissues. So very important molecule. I'm going to get into the idea of Watson and Crick. I'll briefly mention them here. I'm going to upload a video that shows kind of the scientific methodology, the history of the discovery of this molecule. It's quite interesting. So Watson and Crick, we associate them, and there's some drama as well, but Watson and Crick, we associate them with the structural knowledge. They discovered what this molecule looks like, right? The structure of where the parts are and things like that. So DNA, Watson and Crick, genetic information, all of these go sort of hand in hand. So if we think of DNA as a genetic blueprint, it's the genetic instructions. I don't know if you're a fan of Legos, right? But Legos come with a bunch of scattered pieces. If you want to follow the instructions to build the object that it shows on the box, well, you go step by step by step by step. And that's what DNA is. It's a step by step recipe of genetic blueprint that tells the body where to put particular proteins, where to build this. Over here, we're going to build eyes. Over here, we're going to build ears. Over here, we're going to build a stomach. Over here, we're going to build a brain. So that is the genetic information conveyed on the DNA. So DNA holds the instructions, and DNA can transmit the instructions to the next generation. I'm going to use this term boss, right? I know might mean different things to different people, but when I use the term boss, I say DNA is a boss molecule. And I don't know if you've had a job at some point, but there are different types of quote unquote bosses, right? So let's look at this picture here. In this situation, I'm going to say that's a leader, right? The leader is working with the employees, trying to accomplish the mission. They're working hard, leading by example. If I hope you're fortunate enough to, if you work, I hope you work under a leader. Right? It makes life a lot more pleasant, right? It makes the work environment a lot more pleasant. In this situation, we have a boss, right? So the boss isn't actually participating in the work, but the boss is ordering commands. Hey, do this, do that, hurry up, right? That kind of stuff. So DNA is a boss, right? DNA actually doesn't do the work. DNA gives the orders to have the work done, right? So this would be DNA. And who is it commanding? It's commanding the enzymes. It's commanding these little instigators to go and catalyze these different types of reactions. So again, it gives instruction to all parts of the cell, but it doesn't actually get his hands dirty. It doesn't actually do the physical work. It's just giving the instructions, right? So boss, DNA being that boss molecule down here. Now, what are some of the instructions that DNA is going to give, right? And, you know, just going to get a little, maybe PG-13 a little bit here, but one thing DNA is really good at doing is kind of commanding the body, be it a human body, be it a dog, be it a cat, be it a tree, any type of living entity, right? One of the qualities of life, and we're going to get into this a little bit later, but one of the qualities of life is it has the ability to procreate more life, right? So what DNA is sort of in charge of is this genetic instruction that after a certain age, after we hit, let's say, puberty, DNA kind of kicks in and it starts to have an impact on libido, right? Libido is defined as the sex drive. So I don't know if you have like little brothers, maybe sisters, but little brothers that are approaching puberty. You know, 13-year-old little brother, like, man, your little 13-year-old little brother is starting to feel that libido, and man, they get a little bit creepy even sometimes, right? They smell of puberty. They have one thing on their mind, right? So that is genetically programmed, right? Once we get to a certain age, once we get of reproductive age, DNA starts to send these signals. You're reproductive, let's reproduce, so the DNA can be transmitted to the next generation, right? So that's one signal that we're getting, right? Well, to complicate things, DNA also teams up with the hormones, right? We have a surge at puberty, a surge of testosterone. We have a surge of the estrogens in ladies. So those further that sort of idea of the reproductive necessity, right? And I got some random pictures here. Somebody emailed me this a little while back, something like, where do Easter eggs come from? Or this is where Easter eggs come from, right? Well, that rabbit is of reproductive age, and it's getting the signal with its libido, the hormonal influence, and it is, in its mind, it is crucial for it to reproduce. It's not a rabbit, but a chicken is close enough, I guess. I don't know. Right? Here we see a little, let's see, a cat. Well, she had kittens. All right, cute. But as far as DNA is concerned, DNA would say, okay, yes, I'm going to be now transmitted to another generation. And if reproduction doesn't happen, that genetic lineage, that DNA stops being transmitted, right? And that's the that's not good for the DNA per se, right? We're personifying DNA, but DNA wants to be transmitted from one generation to the next. Here we have a picture of a dog. I think that dog is quite skinny. You can see her bones, her skeleton, right? So very, very thin dog there. If DNA was really, how do we say, if DNA really cared about the health of that dog, it would say, no, 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 don't reproduce this cycle. You know, go get you some food, pack on a little bit of weight, be a little bit patient for the next sort of reproductive cycle, right? But that's not what DNA tells the dog. DNA tells the dog, well, yeah, you're skinny, you're hungry, but that's okay. Reproduce and we'll worry about that other stuff later. Right. So in that situation, DNA sort of takes charge of the sort of the thought process, the instincts, these natural biological drives. And this dog, we can tell, has puppies, right? You can see that she's nursing there. I don't know, this next picture. Maybe some of you have gone through this drama, right? This is a life-changing drama. So this young girl in this situation, maybe female, same thing for males. Your whole life ahead of you, full of potential. But hey, you know, if that libido kicks in, hormonal influence, and to make things even more complex, if there's alcohol involved or certain recreational drugs, that's 3 against 1, right? And nature takes its course. And nature doesn't care, DNA doesn't care if she was, you know, she had her whole life ahead of her. Now, you know, she's with child, and now it's, you know, complicating a little bit her life, right? For good or for bad. But that is the goal of DNA, right? And when we cover DNA, we can take it in different ways. We're going to cover the boring biochemistry part, the molecular structure, but DNA can kind of go in this other psychology route, sociology route, and I think that's a lot more interesting, right? And it does have a huge impact on your daily life, on the choices that you make, on things that you're interested in, you know, the orientation of who you're attracted to, all of this coming back to that DNA molecule. And in a sense, we say, well, let me backtrack, right? We know DNA has this ulterior motive of wanting itself to be transmitted at the maybe expense, at the complication of life, that kind of stuff. But there's an individual by the name of Richard Dawkins, Dr. Dawkins. He is a geneticist, sort of sociologist, and he has proposed this idea of what we call the selfish gene hypothesis. Again, this is that idea where DNA is personified. DNA then, according to Dawkins, and this is a hypothesis, not a proven theory, but this hypothetical idea that DNA is looking out for that particular individual. So the DNA in your body is trying to take care of you, right? And it's trying to take care of you first. Maybe secondarily, okay, I care about me, but then maybe I care about my family, or maybe a close friend, that kind of stuff. But you are the primary, according to Dawkins' selfish gene. Your DNA is considering you the priority, and it needs to take care of you. It needs to sort of guide you, instruct you in a certain way, so that it increases the odds, the chance of it being passed to the next generation, right? And in that idea, we say it's selfish, right? In this picture, right, these children aren't sharing, right? Over here, oh, I don't think they're, that, I don't know, that looks kind of a little bit close to fighting to me, right? It started out, oh, it got pretty cheeks, no, and then it got kind of, I don't know, right? But here we see some deer fighting. So why would we generate conflict? Right? And why did you have to be taught to share? Sharing doesn't come natural to most humans, right? Sharing is this concept that you have to give of what you have to somebody else. And we call this altruism, right? So the idea of being altruistic is that, you know, you put your resources, something that you have in abundance, you share with others, right? You even might put your life at risk, your health at risk, you inconvenience yourself to make life easier for others. And there are some very, very altruistic people out in the world, just naturally, but for the majority of the human population, that doesn't come natural. That has to be taught, right? and again, Dawkins gets into the psychology and these ideas. Let's say you're some wealthy individual, right? So you're giving money to help somebody. Well, is that truly altruistic? Or are you giving this money to help somebody and you benefit in some way by having a tax write-off? Or you benefit in some way by generating publicity in some way? Or, there's this idea, man, you can analyze yourself. Like, you do something nice for a person, do you do it in hopes of being told thank you? Or do you do something nice in hopes of them repaying that favor or something like that? So if you really are honest with yourself, maybe you might see some validity to this hypothesis, right? Some of us think, no, I'm truly nice, the core of my heart, right? So, and that may be the case, but a lot of individuals, again, will truly find that, again, you do something nice, the whole basis of religion, right? Why do you do nice things? Well, I do nice things now because maybe later I don't want to go this way, I want to go that way, right? So you do nice things in hopes of getting some nice payback. The concept of karma, right? I do this nice thing today so I can get something nice later. So again, the idea that you don't do something just out of the goodness of your heart for no particular reason, right? That would support this selfish gene hypothesis. If you analyze this local COVID situation, right? So what happened with the toilet paper, right? What did DNA say? Man, there's a virus out there. There's a limited supply of toilet paper. I don't quite understand that link, but somehow, some way, people decided, I'm going to go run out, not just take the normal allotment of toilet paper. I'm going to take enough to take care of me, to take care of my family. The other strangers, I don't care, right? And that really supporting this idea of the selfish gene hypothesis. So again, analyze it if you will, and see if that makes sense to you. But that's the context we're going to continue as we look at this boss molecule, this DNA molecule here, right? So I don't know if you're fans of the movie Deadpool, right? Maybe you've seen it. Maybe you've seen it with your little brother, little sibling or something, right? Well, I think there's a fact that involves libido, and there's also a situation that entails, I guess, this idea, and this goes back to morals, this goes back to culture, to ethics, to our American society, right? This idea that libido is something unnatural, something bad, something nasty per se, right? But in the movie Deadpool, right, there's a lot of violence per se. There's killing, there's stabbing, there's injuries. But I don't know if when you watch this movie with small children, do you cover their eyes when somebody's getting injured? I doubt it, right? But there's going to be some parts where there's consensual sexual activity. And these are the moments when most families, cover little Junior's eyes, right? No, don't look, porque they're doing adult stuff, right? To me, this is kind of interesting, right? Violence, murder, that's actually against the law, right? That's a breaking of the law, right? Consensual sex is not, right? Two consenting adults, that's something very natural per se, biologically natural. So what is it about our American standards that make us, cover the kid's eyes in this scene and not in this bottom scene? Yeah. So it's just things to consider. We know that in the advertising world, the idea that stimulates the libido using sex as I guess, something to motivate the consumers to spend money is well known and utilized, throughout, right? So I don't know how much libido you have, right? How, where your thoughts are, but I see here 3 bottles of a beer. I don't know what you see, yeah? I don't know. I guess this is used these scantily clad individuals to help sell perfume, right? They could have been, you know, in a nice business suit, nice formal attire, but no, that doesn't attract the same amount of attention, right? Wow, okay, this is an interesting link between Mentos and something's happening there. Right? So again, throughout our society, we see this a lot, that these advertisers playing on the idea, catching our interest, kind of making sure that that image, you know, makes us alert and pay attention to the products that these individuals are selling, these companies are selling. So we get into the idea of basically money, right? And I don't know if any of y'all, maybe not this semester, yeah, but in past semesters, if you've ever been out at an ATM late at night getting cash, that cash probably is not going to go to pay the mortgage or to pay the light bill. That money is going to go to kind of try to satisfy the libido in some way, shape, or form. Yeah, and that's sort of the idea where a lot of manipulation can happen consciously, subconsciously. If you've ever bought a drink for a lady or for a guy, right? So did you buy the drink because, man, you know, she just looks very thirsty. I bet I don't want her to suffer dehydration. Let me buy her an alcoholic beverage. Yeah. So I don't think that was just an altruistic behavior. I think there's another, you know, we're doing a certain action in hopes of getting some sort of reciprocal type of behavior, right? So the idea of libido, manipulation, maybe subconscious manipulation, doing certain actions that increase the odds that reproduction may happen. A certain choice in clothing, right? In footwear that doesn't look nothing at all comfortable to me. But ladies, why do you wear that, right? I've seen ladies almost breaking ankles, no? But they choose to wear those. Why? Again, only you know the answer. But Dawkins would say, well, the DNA somehow has manipulated, had an impact on the libido, and throughout, I guess, modern history, we associate footwear like that as being attractive, sexy, maybe increasing slightly the odds that that a certain someone, you know, you're going to catch their attention. Certain stores, certain companies making millions of dollars, billions of dollars probably, in selling certain types of clothing. These individuals, man, it looks cold, right? But wow, well, why are you out there with such little clothing, right? So again, what would we say there, right? So instead of having long, you know, big coat, warm pants, that kind of stuff, what choice of conscious or subconscious decisions took place in order for her to venture out into the street like that, right? So, and it goes both ways, males, females. But what typically happens, DNA allows for some very fun experiences, but may have some long-term consequences in terms of maybe, you know, generating new life or you know, transmitting certain types of STDs, that kind of stuff. But hey, you only live once. That's the DNA mentality, and you know, live it up, right? And that's, again, has a big impact on sociology. In this situation, we analyze the idea. I've always thought this is interesting, that in the United States, we're quite poor at math collectively, right? Most Americans are not good at math compared to other countries, Japan, China, India. My hypothesis is that we do have the aptitude for math. However, most of the basic fundamentals of algebra, trigonometry, are taught sort of between like 7th and maybe 9th grade. And if you analyze those grades, that's when these hormones are raging. And that's where a lot of our focus is not on the quadratic formula, right? It's not over here on algebra and trig, but it's, you know, kind of somewhere else, right? So this is the idea that we're presenting these hard concepts at an age where a student's mind is not really focused on these hard concepts. So they're distracted. Hey, pass this note to this person. Pass this note. Oh, you know, you want to go out, that kind of stuff. So really having an impact on that subconscious idea, subconscious behavior, subconscious focus, that kind of stuff. This individual, right? Well, looks like in a intimate situation here, but as she's passing, what happens, right? What does the DNA say? Well, just, you know, it sometimes doesn't turn off when it's supposed to, right? That's sort of the idea of being a civilized adult, right? Being able to control some of these urges and not let the urges control the adult individual, right? And again, a lot of sociology that this will carry off into. We're not going to necessarily continue down that route, but this is an introduction for these concepts later on in your careers. I don't know how religious you are, right? But once you walk into those church doors, those synagogue doors, that holy place, this libido signal doesn't just automatically turn off. I'm in a holy place, let me focus on holy stuff. That doesn't quite work that way, right? Some of you, you go to that holy place and you have impure thoughts. Oh my God, this person is, thank you, Lord, right? You start to have these thoughts that they just don't turn off, right? And that's sort of the idea of libido, right? It's constantly there, that little voice. He's attractive, she's attractive. Ooh, yeah. Ooh, I want to hold hands. I want to go hug this person. Right? So that's the idea of DNA, the selfish gene hypothesis, this ulterior motive of always wanting to be sort of in a situation where it can increase its chance of being passed on to another generation. So manipulating the human body for its own motive of continual transmission there. Yeah? So I hope all that made sense. Now, again, we're going to kind of hop back to that sort of sociology stuff when we get into genetics and some other discussion. But let me come back to now the boring discussion on biochemistry. So we talk about nucleic acids. Nucleic acids are this big boss molecule, but they are made up of small units, right? That's the definition of a macromolecule. The little small units, the monomers for nucleic acids, are what we call nucleotides. So nucleotides are a little 3-unit structure that looks something like this, right? So we bring back one of our functional groups, right? If you recall a functional group from chapter 2, the phosphate functional group, PO4. We bring back the concept of a base, something alkaline, a pH above 7, right? So we have a phosphate group. We're going to have a nitrogenous base, and we're going to have a carbohydrate in this situation. So when we have these 3 units connected together, we have a 5-carbon sugar, a phosphate functional group, and a nitrogen-containing base. Base plus sugar plus phosphate in this orientation. The base connected to the sugar, connected to the phosphate. Collectively, these 3 things we call one singular nucleotide. Right? And a bunch of nucleotides together makes up this thing we call the nucleic acid. So we have a few options, right? We're only going to have actually one phosphate. We can't change this molecule at all and still call it a phosphate. So we only have one type of phosphate possibility, but we have 2 types of sugars that we can have. Right? We can have this thing called a ribose with a hydroxyl group on carbon 2. Again, 5-carbon sugars, kind of hard to see, but we have carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. On the carbon 2, we have a hydroxyl. On this other molecule, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. On carbon 2, there's no oxygen. So the hydrogen is just directly connected to that carbon. So if we have a ribose and we deoxygenate ribose, we're then left with something we call deoxyribose. And this is, again, the two options of sugars we can have. We can have a ribose sugar or a deoxyribose sugar. So again, one phosphate, we have the potential of a ribose or a deoxyribose sugar, right? So we have two options for the sugar depending on what is coming off of carbon 2. For the bases, it's a little bit more complex. We have two families of nitrogenous bases, right? We have something that we call the pyrimidines. Pyrimidine basically refers to the geometric shape. I think you know a triangle, a square, a parallelogram, that kind of stuff. Well, in biochemistry, this sort of six-sided honeycomb shape with a with a carbonyl coming off here, hexagonal shape, honeycomb shape, carbonyl, hexagonal shape, honeycomb shape, carbonyl over here. If we have this particular shape, biochemically they call that a pyrimidine. So pyrimidine refers to the geometric shape of that structure with that carbonyl coming off to the side there. So we can have pyrimidines in that make up, that compose that base. Or we can have what we call a purine. So a purine is this shape. It's kind of like a five-sided figure connected to a six-sided figure. A five-sided figure connected to a six-sided figure. So that shape, that 5 and 6 unit connected, the 5 and 6 unit connected, we call that structure geometrically a purine in biochemical vocabulary. So we can have either a pyrimidine or a purine. So again, we can have either the pyrimidine shape or we can have that five-sided and six-sided figure, right? So we have two options of pyrimidine or purine. Again, it gets a little bit more complex. If we have a pyrimidine, we can have the specific pyrimidine that we're going to call cytosine, and they signify this with the letter C. So don't get confused. Don't confuse that C with carbon, right? There is carbon in cytosine, but the whole honeycomb shape, that is the actual pyrimidine, right? So we have the cytosine, the specific cytosine base that is a type of pyrimidine. So again, we have the cytosine base, which is that six-sided figure with the carbonyl that falls under the pyrimidine category. We can have a different details, but we have a different type of pyrimidine with a little bit different composition that we're going to call thymine. And we have a third pyrimidine with still a third structure that we call uracil. So cytosine, thymine, and uracil are all pyrimidine shape. And it doesn't work completely, but may help a little bit. If you notice, I have a Y. So there's a Y in pyrimidine, there's a Y in cytosine, there's a Y in thymine. Uracil doesn't have a Y, but it's not too far from Y, I guess. I don't know. Maybe that makes a little bit of sense. So if we're looking now at the purine shape, there's two types of purines. Something we call adenine A and guanine G. So two types of purine structures, three possible types of pyrimidine structures. So again, this is something that you'll have to, distinguish, differentiate on the exam, on the homework. So again, I hope that's making a little bit of sense. The physical geometric shapes of these molecules, the position of certain functional groups is important as well. There's an amino, there's a carbonyl, the amino's over here on this one, the amino's on the top, there's no amino on this one. So that's how they get the different distinctions there. But So to summarize, right, we have to have some sort of base. It can be either a pyrimidine, the pyrimidine shape, or it can be a purine shape. And you will know if it's a pyrimidine, it can be a cytosine, it could be a thymine, or it could be a uracil. Or we can have a purine. If it's a purine, it could be an adenine, or it could be a guanine. Once we have that base, we then have the option of connecting it with either a ribose with the hydroxyl group on carbon 2, or a deoxyribose with just the hydrogen on carbon 2. So we have a base connected to a sugar, and to complete our nucleotide, we bring in our phosphate functional group. So the phosphate bonded to the sugar, bonded to the base, is what we call one nucleotide. And that's, again, the foundational monomer for these more complicated polymers that we call nucleic acids. So again, this is generic phosphate sugar base. Let me give you more specific, more realistic detail here. So if you notice down here, I have a phosphate connected to a deoxyribose. There's no hydroxyl here. And then I have a two-sided, five-sided, a pentagonal shape and a hexagonal shape. So I know that's one of my purines. Specifically, that is guanine. Right? So I have a phosphate plus a sugar plus a base. That equals one DNA nucleotide. Now, how do we know it's DNA, right? I'm going to explain that in a bit, but for now, just phosphate, sugar, and base. Over here, I have another one, phosphate, sugar, and base. Phosphate, sugar, and base. Phosphate, sugar, and base. So what enables this nucleotide, the second one from the bottom, to connect with this first one from the bottom? Well, that connection is a covalent bond between the phosphate of one and the sugar of the other. It's a covalent bond. It's simple. It's a covalent bond generated by that process that we call dehydration synthesis, right? So dehydration synthesis forms a covalent bond. However, the geneticists like to sound very educated, and they like their own vocabulary. So they give it a different title. They're going to call this a phosphodiester linkage. It's the same thing. It's a covalent bond, right? But they call it a phosphodiester linkage, right? So it's connecting the phosphate of one nucleotide to the sugar of the other, right? So between sugars and phosphates. And what ends up happening, we have a very repetitive type of organization, a repetitive type of structure. We have a phosphate connected to a sugar, connected to a phosphate, to sugar, phosphate to sugar, phosphate to sugar. And generically, we then sort of refer to this as a backbone, with the analogy that the human body has the vertebrae, the backbone that runs down your back, and then the ribs kind of come off of that backbone. So here we have our backbone, and then extending, protruding out, we have our ribs, or our actual bases there, right? So that's sort of the complexity. We're starting small, and we're adding more and more and more, building more complexity to our DNA molecule. So once we have completed our molecule, once we've completed our DNA molecule, our deoxyribonucleic acid, there's a couple of details that are important here. DNA is going to include 4 of the 5 possible bases. Right? So let's come back over here. Sorry, backtracking. If you remember our 5 bases: cytosine, thymine, uracil, guanine, adenine. 4 of these sound kind of similar, right? Cytosine, adenine, thymine, guanine, uracil. It just sounds weird, right? Uracil doesn't even have a name that kind of matches the others. So uracil is not quite cool enough to be in the DNA club. So DNA will only include 4 of the 5 nitrogenous bases, right? It contains 2 of the pyrimidines, cytosine and thymine, and then contains the 2 purines, guanine and adenine. So when we talk about DNA, we don't talk about ribose sugar, we talk about deoxyribose sugar. So DNA, right? Deoxyribose sugar. When we build our strand, right? Let's say our phosphate sugar, phosphate sugar, phosphate sugar. Here's our sugar phosphate backbone. In this situation, our bases are protruding outwards. We're going to have a complementary match on the other strand. So we don't have just one strand of DNA, we have 2. And they're going to be running in opposite directions. So is what we call a anti-parallel strand. They're parallel, but they're going in opposite directions. There's a direction, a trajectory. In physics, we say there are different vectors, right? They're going in different directions. So deoxyribose sugar, 4 of the 5 bases, anti-parallel strands, and again, our directional terms are going to be important here. When we talk about DNA, we can't say top or bottom, left and right. The molecule is all twisted. So to know which direction we're talking about, we talk about the 5' end and the 3' end. Again, let me backtrack. So when we look at our carbons on our deoxyribose and ribose as well, but right now we're focusing on deoxyribose. If my deoxyribose is right side up, we notice carbon 5 is closer to the ceiling than carbon 3. Carbon 3 is closer to the floor. Right? Now, if you can envision this deoxyribose turned upside down, then carbon 3 would be closer to the ceiling, carbon 5 would be closest to the floor. So that's where we're getting this notation. So notice here, carbon 5 is higher, carbon 3 is lower. If we're running anti-parallel over here, we must have the opposite situation. So here, carbon 3 is closer to the ceiling, carbon 5 is closest to the floor. And you can see kind of here we're pointing upwards to 5, here we're pointing downwards to 5. So they're going in opposite directions, they're anti-parallel. If you notice the bases, they show them upside down. The phosphates are it's not a letter D, that's a phosphate, trying to indicate the anti-parallel nature of this molecule. Another piece of the puzzle, we're going to complicate things a little bit here. Well, maybe not complicate, but we're going to put another sort of law in place that we have to address. And we're going to call this the complementary base pairing law. This is a more modern name. The old name, the name I learned, is we used to call this Chargaff's law. Right? This is Erwin Chargaff. This is that biochemist that discovered this fact about DNA. But according to Chargaff, he established, once he studied DNA, he found that there must be a large purine connected to a smaller pyrimidine. And he found that this was always the case, right? When we back up, when we form our double helix, or when we haven't talked about the helical nature, but this anti-parallel strand starts to spiral. This one starts to spiral like a protein, like an alpha helix, but it's going in the opposite direction. I can't quite do that move, but we're going in an anti-parallel type of motion there. Right? But if you notice, if we use the analogy of a ladder, so this sort of vertical support structure of the ladder allows the little cross perpendicular, handholds and footholds there, the rungs of the ladder. So if I have a particular long base, in order to keep this parallel, I need to compensate with a short base. If I had a long one here and another long one, it would cause the ladder to bulge out a little bit. Right? But what if I had a short pyrimidine bonded with another short pyrimidine. It would cause the latter to kind of, cave in a little bit there. So we don't want that. We want parallel. So to accomplish parallel, I need a large purine connected to a smaller pyrimidine. Large purine to a smaller pyrimidine. Large purine to a smaller pyrimidine. Large purine to a smaller pyrimidine. But we have one more level of detail here. Right? Anytime we have the large adenine has the potential to form 2 hydrogen bonds with thymine, and that's the most comfortable configuration that they can achieve. So you're always going to see in the DNA molecule the adenine bonded with thymine, and it always reminds me of AT&T, right? A always connected with T, adenine always with T, thymine always with adenine. Right? It's always the case. If you know that vitamin store, that nutrition health store, GNC, that reminds me of G and C, right? Guanine and cytosine. So when we have guanine has the sort of the molecular shape structure that enables it to form 3 hydrogen bonds, as does cytosine. So these two are going to fit most comfortably together, whereas these two are going to fit most comfortably together. So adenine always with thymine, guanine always with cytosine, right? And these are the what we call Char-Gaff's rule. Now, we go back to chapter 2. What holds these 2 bases together? Well, those little red dotted lines, we know these are hydrogen bonds. We have a delta positive on hydrogen, delta negative on oxygen. Delta positive on hydrogen, delta negative on nitrogen. So because we have opposite delta charges, these 2 bases can attract together and form those hydrogen bonds. Same thing here. So again, hope you can visualize the shape. The structure is going to be important to understand how it functions later. Right? But we have our sugar phosphate backbone. Over here, we have a sugar phosphate backbone going in the opposite direction. We're going to have deoxyribose sugar. We have our nitrogenous bases held together by hydrogen bonds, and we have again the purine connected to the pyrimidine, specifically the adenine connected to the thymine, the purine guanine connected to the pyrimidine cytosine. Right? And these are, once you know that, I mean, you know the molecular structure of DNA. It just repeats and repeats and repeats and repeats. Now, the video that I'm going to upload after this discussion gives us a little history, a little soap opera, if you will, on how the DNA molecule was discovered. Right? So it did involve the scientific method. There were some incorrect hypotheses that came first until eventually they got it right. And there's a couple of different examples of good collaboration and maybe some competition. There's some examples of not good collaboration, right? So as you watch the video, kind of look at the relationship Watson and Crick have, the professional working relationship that they have, their collaboration, that kind of stuff, right? Analyze the sort of the relationship, the professional work sort of collaboration or lack thereof of Wilkins and Franklin, and see what kind of role Pauling has, right? And then that's an important date. We're gonna, after you watch the video, next slideshow, I'll kind of come back and address that. But an important date, February 28, 1953. Most biologists understand the significance of that date there. So this is the video. I'm not going to show it here. I'll upload it onto Blackboard. You can see it, after you're done with this slideshow. And this will make sense more when we cover that. All right, so I'm going to stop the slideshow now so it doesn't get too much bigger. I'll stop it now. You can process that. Then I'm gonna sort of get into this idea of forensics. How do we apply our DNA knowledge in modern forensic science, right? So I'll get into this slideshow in a bit, but we'll conclude this one now. Let it convert and load. Normally takes a little while here. So again, I'm back. I'm ready to really start recording lectures and catch up as quickly as I can. So hopefully this made sense, and keep an eye out for the video, keep an eye out for this lecture and for the lectures that follow.